So up next we have Yichen. She's a postdoc at the Wendy Schmidt Center, and she'll be giving a talk about segmentation-free analysis of submicron-resolution spatial transcriptomics.
Right. We've heard about spatial transcriptomics in this section. So spatial transcriptomic technologies have been developing rapidly in the recent years. Two of the key aspects of those technologies are the spatial resolution and the number of genes profiled. So imaging-based technologies can measure the precise location of every molecule, but can only profile a pre-designed gene panel. And commercial platforms are expanding their throughput, scaling from a few hundred to thousands of genes. Sequencing-based technologies, on the other hand, can unbiasedly profile the whole transcriptome. But earlier technologies have rather low resolution at a multicellular scale. So more recent technology like 6-scope and stereo-seq have achieved submicron resolution comparable to microscope. So our methods are designed for any spatial transcriptomic data that with subcellular resolution. So the data from such technologies are spatially resolved transcripts. Each pixel contains one or a couple of transcripts. Here the color representing different gene identities. So such high resolution and high throughput data does not automatically enable high throughput inference. A basic task of data analysis is to recover cells and their cellular functions. The current standard approach is to use nuclei staining and cell boundary markers to perform cell segmentation before doing single-cell level analysis. However, high-quality imaging information is not always available, especially for some sequencing-based platforms. So even when cell segmentation is feasible, it's still a challenging task. Since we are making a two-dimensional section from a three-dimensional tissue, the cross-section of a cell is often of irregular shape, and some cell nuclei may not be captured in this 2D view, and they may be missed from downstream analysis. So there are more technical difficulties in practice that jointly result in imperfect cell segmentation. An alternative way is to do grid-based analysis, to collapse those sparse information from individual pixels into bigger units enabled to do statistical analysis. This approach can use all the transcript, but it loses spatial resolution. And often, such a grid contains a mixture of different cell types. The ideal solution should annotate cellular function at each individual pixel and do so efficiently for large tissue regions. And this is what we aim to achieve. So here's the intuition behind our method. So we start from a hexagonal grid and infer each hexagon as a mixture of multiple latent factors representing cellular functions. These factors can be learned de novo from the data, but we could also incorporate prior knowledge or use external single-cell reference panel. We call the center of the hexagons as anchor point, and then we slice this grid to generate a denser lattice of such anchors. But at this stage, each anchor still represents the mixture. So the key step is to allow each pixel to look around and pick which anchor it goes to. And then the anchors will collect those pixels and update their factor proportions. When this process converges, we have a probabilistic factor assignment of every single pixel. We will use public data from 4 different platforms to show that our method works in practice. But we do emphasize that we work for data from all such subcellular technologies. Right. Stereo-seq is a next-generation sequencing-based technology. This is a 16-day mouse embryo section. Over 750 million transcripts are captured, but only 40% of them are assigned to any cell. Because on this platform, only nuclei staining was available, so cell segmentation is very challenging. On the left is the published cell-level annotation. On the right is our unsupervised pixel-level analysis without using the nuclei information. We recover similar global structure, but with additional details. One example I want to show is the developing muscle tissues. So during myogenesis, myoblast matures into myofiber. Cell segmentation is particularly difficult for the later stage with these multinucleated fiber-like cells. The top right panel shows nuclear staining, and the bottom is a published cell segmentation-based analysis colored by annotated cell types. We cannot see mature muscle fiber in this view, and almost all the cells were labeled as myoblast. However, when we applied our method, we see the emerging striated muscle cells. The top image color each pixel as our inferred latent factors, and the bottom highlight two factors, the red pixel coming from myoblasts and the yellow pixel belonging to differentiated muscle cells. So imaging-based platforms often have high-quality imaging with multiple channels to support better cell segmentation. So let's look at the muscle tissue at this mouse pulp data from Xenium with 5,000 gene panel. So in the middle is the image with nuclei staining and cell membrane markers. On the right is a standard cell level analysis. On the left is features output. The color here also represent the inferred latent factors, and we label them by the signature genes. The purple background here has the signature of immature muscle fiber. The yellow marks the thin fiber-like structure located between muscle cells that has the signature of fibroblast. Another type of intermuscle signal we see is from endothelial cells in green. So, well, in this data, the cell segmentation is quite good, but still the fine-scale tissue structure is lost. Right. The next example is from brain tissue. Here's a mouse half brain with from Cosmics. Again, there are almost 40% of transcript were lost in single-cell level analysis. Let's look at one such cell sparse region near hippocampus. So starting from this granule cell layer, we have three distinct molecular layers that are filled with dendrites and axons, but does not have dense cell bodies. Between the dendritic layer of hippocampus and the cortex, we have the white matter tract that are dominated by myelinated axons. The cell body inside this layer are indeed myelinating oligodendrocytes. Right. In the cortex, we also observe a different expression pattern near nuclei versus other part of the cell. Here, the green factor here often appear at the center of the cell body, is enriched for two long non-coding RNAs retained in nuclei. And different neurons may share this nuclear signature while having distinct dendritic or synaptic activities. We checked another public brain data from Merscope to see if we observe similar subcellular pattern in a different technology and from a human instead of mouse. Especially in this case, those nuclei markers are not available in the gene panel. So although the nuclei non-coding RNAs are not present, the model still seems to learn a soma-specific signature. The signature gene of the red factor encode neurotransmitter receptor and voltage-gated channel subunit that are predominantly located in the cell body. And the pink and yellow are both enriched for mRNAs targeted for dendrites or presynaptic channels, terminals, and they seem to be from two different subtypes. Right. Another major application of spatial omics data is cancer biology. So here is a human breast cancer tissue from 10x Xenium. The released data includes cell segmentation based on nuclei. We will zoom in this region around the ductal carcinoma in situ. So the top right histology image shows adipose tissue. The empty spots are created by large lipid droplets in adipocytes where no RNA is present. The nuclei of adipocytes are much smaller than the cell body and located at the cell boundary. Often there are immune cells that are much smaller and located between adipocytes. The cell segmentation from Xenium from 10x platform is quite misleading in this region because nuclei staining is just not very informative. So our approach is not affected by such cell morphology. The top right panel shows color each pixel by our inferred factors. The bottom one, we highlight adipocytes in yellow and endothelial cells in blue. We also identify different immune cell population in this region. So overall, we provide a more realistic picture of the underlying biology. Right. In breast cancer, cancer-associated fibroblasts and extracellular matrix are major components of tumor microenvironment. and are important for cancer progression. So here we use another public data from breast cancer tissue immersoscope to show the potential advantage of our approach in this case. On the left, the white pixels shows the actual cellular matrix building-related signature. They are in the region also rich in tumor-associated macrophage. On the right, we see the co-localization of these two factors in a different context. The light green pixel, with this similar fiber-like structure, are also cancer-associated fibroblasts, but it has a different functional signature. So if we look at the image, this region is actually cell sparse. Those fibroblasts does not seem to be well captured by cell boundary markers. Comparing with cell-level view, our approach is perhaps better suited to study tumor microenvironment in a more unbiased way. Right, so not only does picture provide biologically meaningful inference across platforms, but it's lightweight and highly scalable. So here we compare with two representative methods. Beta has been, one of the most accurate pixel-level analysis methods, but it's not designed for for current high-throughput data. And the memory requirement is prohibitive for even a fraction of the state-of-art dataset. The GraphST is a method that uses popular graph neural network. It works at lower resolution and all require segmented cells. But still, the projected cost for analyzing large data is quite huge. So we have adjustable memory budget that's independent of the size of the dataset. In terms of runtime, we scale linearly with the number of transcripts and can be massively parallelized. Beta scales superlinearly and cannot scale to more than 1,000 genes. And our pixel-level inference is even more scalable, efficient than the cell-level inference from the graph neural network. So in summary, we present a segmentation-free analysis tool that infers cellular function at submicron-resolution. It applies data from any subcellular level platforms. It's orders of magnitude more efficient than existing method, and is built for future large-scale multi-sample dataset. And it does not rely or limited by imaging-based cell segmentation. We just made public a new version of our software, and we are actively working on new functionalities to support different use cases. I'm looking for collaboration and maybe looking for a job soon. With that, I'd like to thank all the wonderful people I've worked with and happy to take any questions.
We have time for one or two more questions. So I guess as the spatial technologies get like finer and finer resolution, do you foresee this kind of continuing to be a question of doing this in a cell-free manner versus segmenting? Like going forward with the way spatial technologies are developing, how do you see this being used?
I'm not quite understanding the question. Spatial resolution is proving, but there's a physical limit of what you can measure, because after some point, for imaging-based platform, you have this optical crowding issue. For sequencing-based, we are doing slightly better, but still, there's a lateral diffusion of transcripts. So I'm not entirely sure how much we can improve upon current technology in terms of resolution.
Thanks. Okay, let's thank Yichen one more time.